All right, here we go. We are dismantling the critics on endogenous retroviruses. Solo LTRs, are they degenerated ERVs or functional stretches of DNA? And we're just going to get right into it. I want to make sure that the audio is shared, though, because I will be starting us off with a video, just a clip from a video between Dr. Stern Cardinale and Dr. Fuz Rana, where they were briefly discussing ERVs. And Dr. Dan, he has, in this clip, put forth a couple arguments that I wanted to address. Arguments that comprise evolutionary philosophy, which Dr. Dan is so good at, as evidenced by a recent discussion or impromptu debate I had with him on the chromosome 2 fusion. So he definitely has earned a PhD in evolutionary philosophy, storytelling, and imagination. And I am going to provide yet another example of that here. So I'm going to mute. We're going to play the video, and I am going to go over some slides on it. Here we go, guys.
So this gets at something that I'm glad you brought that up, because this gets at something that I think I've read from you. You've said that we have strong evidence of I'm paraphrasing. Correct me if this is not an accurate representation, but I think you've written that we have strong evidence of function in every class, in every major class of DNA element, where you could go
Right.
ERVs, signs, lines, whatever, and you could say, here's an example that we have really strong evidence does a particular function and is selected to do that function.
Right.
And I think you're absolutely right when you say that we have strong evidence of function in every class of DNA. But where I get off the train is, okay, we have evidence of some members of that class, but if a class of DNA, endogenous retroviruses are a great example, because that's 8% of the genome. There's like 200,000 of them in the human genome, but only about 20 are the full, like, LTR, gene, LTR, like the complete structure. And the rest are degraded in some way. And most of them are either just the paired LTRs or just solo LTRs. Many of them, if they're transcribed at all, it's very little. And most of them aren't. They're just methylated and go and sit in the corner and don't do anything. And so you could have a situation where you have retroviruses that have been co-opted for this very important function. And I think there's super strong evidence that without retroviral endogenization, mammals would not have evolved the way we did. I think there's super strong evidence for that. But I don't think you could extrapolate that to most of that 8% of the genome that's retroviruses, just because the ones that are doing the activity for which we have, I think we would agree that they're functional. The ones that are doing that don't look like 99% of those sequences in the genome. And the rest of them, it's not just that we don't have evidence that they don't do that thing. It's that structurally they can't be doing that thing. They just don't have the parts to do it. And I think that concern works across the board when we talk about like ALU elements. Well, okay, there's some of them that are, in exons and are clearly have been co-opted as part of the actual coding sequence of a gene. But then most of the rest aren't actually transcribed at very high levels at all, if they're transcribed at all. So again, they're not doing that thing and they're not translated. So like, that's where you could argue, yes, there is, I think, strong evidence of function in every class of DNA, but extrapolating that to a majority of the DNA sequence as a whole is challenging.
Yeah, and I think that's a fair point. And that's, at this point in time, that's the, I wouldn't say that's the limitation of my particular argument. And I, and I'll say this, that when I started working at Reasons to Believe, one of the
Okay, so I am going to stop it there and make a few comments. And so we'll get right into this. go through some slides. I just recently did about an hour and a half video on ERVs. If you do check the description box, you will find a lot of the content that I have done on this topic, and there's a lot. I've written an entire book on it. I've done many debates on it, including debates with some of the critics. And you'll find lectures and presentations I have given. And so I just recently gave a presentation, again, about an hour and a half long, comprehensive, talked about the reverse transcriptase and integrase and basically the nature of not only endogenous retroviruses, but retroelements in general. And retroelements do make up roughly 42% of the human genome. And as Dan pointed out, we do find incredible functions that basically these evolutionists like Dan, they will just ignore in terms of how these functions came about. They'll just kind of throw out co-option with no actual evidence. It's an example of uninformative gloss. It's storytelling. If viral DNA, harmful viral DNA randomly inserts itself into the cell of a host, it's more likely to disrupt the functionality of that host or the cell than make it better. And Dan himself has admitted in the past, I have a clip that I'll show a little bit later, that it's never been observed that an exogenous, a harmful infectious exogenous retrovirus like HIV has infected horizontally and managed to infect in the right way in a germ cell line and be passed on vertically to the next generation, and then be hit with the necessary series of mutations basically to wipe away any harmful attributes or effects of the newly endogenized retrovirus, and then evolve, whether through co-option or beneficial mutations, a function that actually without, as Dan pointed to, mammals, as we know, would not exist. And I've shown papers where they admit that without ERVs, a specific type of ERV, mammals would not exist because we require many of these ERVs for placental and embryological development. Now, these retroelements, they are broken up into different classes. One of those classes are endogenous retroviruses, which evolutionists assume are the ancient remnants or vestiges of past viral infections. Now, when it comes to your signs, your lines, your ALU sequences, LTR retrotransposons, non-LTR retrotransposons, we have amazing evidence for functionality in all of these different classes that the evolutionists can't explain, and they admit it's never been observed. But that's not necessarily the point I want to focus on. I want to focus on this claim that we really only have several full ERV sequences, right? Where you've got the LTRs on either side, which stands for long terminal repeats, your GAG, your POL, your ENV components of the ERV, and obviously host DNA on either side. That makes up your complete ERV or endogenous retrovirus unit. The great majority of quote unquote ERVs in the genome actually represent just pieces of retroviral like DNA called solo LTRs. And you'll notice here on the excerpt from this, I would say, must-read paper, intriguingly, almost 90% of all HERVs, human endogenous retroviruses, represent so-called solo LTRs. And so that means that the vast majority of so-called ERVs do not comprise the entire ERV unit. And so what they'll argue here, notice these HERVs lost the prototypical retroviral genes GAG, POL, and ENV. Now remember, the endogenous retrovirus, it has that classic retroviral design. And we talked about this in the last lecture, where I pointed out that retroviruses require a host to replicate. And there's a paradox. There's an unsolvable problem for the evolutionists who want to reject what a lot of creationists, specifically myself for sure, propose as being the origin of retroviruses, which is from the host cell, the host DNA itself. Because what came first, the host or the retrovirus? Well, the retrovirus requires a host to replicate. And so if retroviruses, exogenous retroviruses came from the host, then of course they are going to resemble or share that DNA similarity with ERVs, mainly the fixed ones that were created in the first place. But anyways, what we see in an ERV is we have, again, host DNA duplicated on either side of the provirus when it's actually integrated that way. We have the two LTRs, a GAG, a pol, and the ENV, which stands for envelope protein or component of the ERV. But again, the majority of what we see in terms of these retroviral-like units are solo LTRs. And therefore, the majority of what we see in terms of what the evolutionists call endogenous retroviruses are DNA sequences that don't even resemble full ERVs. Again, that comprise your LTRs, GAG, pol, and ENV genes. And so the majority of the so-called ERVs, again, they lack these components and simply represent long terminal repeats, your LTRs. But, and Dan points this out as being an issue for the design hypothesis, when in fact these solo LTRs actually serve as binding sites for initiating or enhancing gene expression. They're functional, they are important. And it is the evolutionist that assumes that these are degenerated ERVs, older ERVs that have had time through mutations, recombination to basically lose those components. As you can see here, due to homologous recombination of their flanking LTR sequences, leaving single LTR promoters in the genome. Due to their activation upon immune stimulation, ERV LTRs have already been termed landing strips for inflammatory transcription factors. And notice this, and evidence for their role in regulating cellular immune responses is growing. It's growing. Again, we are just in the infancy of understanding the DNA language. We are finding out more and more about these created units of DNA function like ERVs. But again, your full ERVs, your ERV unit, they function in determining cell types, gene expression. They act as antiviral protectors. They function in the placenta and in the embryo. And the evolutionists have no explanation for how these functions could have come about other than evolutionary philosophy, which Dan has a PhD in, which is why he dodges questions. And the answers he provides, he thinks, are based off of sound empirical scientific data, and they are not. It's pseudoscience. It's pseudoscience. It doesn't belong in a textbook. It doesn't belong in a lecture. And again, the evidence for their role in regulating cellular immune responses is growing. The LTRs, your solo LTRs, it's the evolutionists, again, that assume that these are ERVs. I argue we shouldn't even call those ERVs because it's the evolutionists that are assuming that they're older ERVs because they've had time basically to degenerate. Okay, so this tells us that what? The full ERVs have amazing functions, as I just went over, and so do the over 90% of ERV-like sequences. And so again, we share your full ERVs, most of them, with what? Your chimpanzees. They actually form a hierarchy. We see nested hierarchical organizations within primates. But guess what? Since those have amazing functional roles, then of course they're there for a purpose, for a functional reason. They are representative of the functional requirements and shared functional requirements among these different organisms. And the differentiating line of evidence is the function, and it's the function that the evolutionists can't deal with. And they need to tap out. And although they haven't, I think it's pretty obvious to both sides, especially ones that are watching objectively, that the evolutionary community, Dan Stern Cardinale here, they are not actually addressing the discriminatory lines of evidence. Okay? we understand that your ERVs share sequence similarity to exogenous retroviruses. But we've already explained why that is the case, and I will cover that a little bit more later. But the point is, why are we calling these solo LTRs endogenous retroviruses when they may not even represent endogenous retroviruses? And probably based on the data, this is the data. But remember, when you're reading these papers, you need to chew the meat and spit out the bones, because you will get a lot of evolutionary philosophy mixed in with the good science, the good data, such as when they suggest co-option with no evidence to back it up, or simply say, well, these solo LTRs, there's evidence that they are necessary to sustain healthy life processes in the cell, specifically here in the immune response, but they are degenerated ERVs. I mean, that is not based on sound science. And so for us as creationists who are looking at this objectively and utilizing critical thinking, we're looking at the data. And so what the data suggests is these solo LTRs represent functional stretches of DNA. And so no, it's not a weakness or a limit in our model, specifically the young Earth creation model, which I think best explains the origin of retroviruses and the origin of functional retro elements and retrotransposons in general. So no, it's not a weakness. We actually have a great explanation, since these solo LTRs are there to what? Regulate gene expression, to act in the immune responses of specific organisms. Pretty simple, right? It really is. Notice this. Functional stretches of DNA. The endogenous LTRs, again, your long terminal repeats, mentioned above, all act as enhancers and may increase antiviral gene expression in response to viral infection and or IFN stimulation. So we have these thousands of functional stretches of DNA. We also have these ERV units that comprise, again, LTRs, GAG-POL, ENV components. And they all contribute to the function of the biological organism. And they act as antiviral protectors, just like our computers are built, or we can add onto the computer, basically download an antiviral program, which is necessary. It's there. And if a virus comes in, it detects it, it can destroy it. That's what's going on in the genome, is a lot of these retroviral-like sequences or DNA units are there, and they are ready to respond to viral infection. This is evidence of forward thinking, which points us back, as I like to point out to the forward thinker. Here's a couple more papers. LTR function. So firstly, ERVs, they can act as promoters, enhancers, or transcription start sites for gene expression. It's function after function. And a lot of their functions depend on their sequence similarity to exogenous retroviruses. None of these functions, some of them absolutely necessary for life to even exist. And the evolutionist has no explanation for how these functions came about. So here's a couple papers. These evolutionists, they're out of date. They're out of touch with reality in the scientific literature. Endogenous retroviral LTRs as what? As promoters for human genes. And so yes, we have noticed how 90% of ERV sequences are not actually true endogenous retroviruses. Now, the true endogenous retroviruses, yes, they exist, but not in the amount that solo LTRs exist, which exist in the thousands. And so it's the evolutionist that calls those endogenous retroviruses, degenerated endogenous retroviruses, when there's no evidence to suggest that. And they represent functional stretches of DNA that are shared across species, yes. If you want to specifically focus on your great apes and primates, specifically chimpanzees, bonobos, gorillas, orangutans, then yes. They're there for a reason. This is evidence for common design. And if the evolutionists want to argue against common design, it's time to step up. It's time to play ball, and it's time to demonstrate empirically. Show us a technical paper that's not just storytelling or just hand waving away the functions by saying, well, these were co-opted or these are engines of evolution. I've heard it all. I've seen it all. I've read it all. Let's see the empirical evidence. Notice this: endogenous retrovirus long terminal repeats as ready to use mobile what? Promoters. And so here's a paper. Again, I highly recommend reading this. Long disregarded as junk DNA. A lot of these papers you read, this is 2021. And I've got papers in my book from 2022, and the evidence is mounting. And, which means the evolutionists have to get better and better at storytelling since they can't address the empirical evidence. And you'll read these papers, and I've read a lot of them in especially the preparing of my book, but doing debates on this topic, lectures. And sometimes you'll read these papers and they almost sound like they could be written by an intelligent design proponent or advocate. Long disregarded as junk DNA, they admit that ERVs and retro elements were once assumed to be junk DNA. So they did not, but you hear people like Dr. Dan, Stern Cardinale, Andrew Cumming, and Speed of Sound on their Answers in Atheism channel gave a response to my last video. I think they only played like 10 seconds of my video, misrepresented a lot of what I'm saying, ignored 90% of what I'm saying. And someone like Andrew Cumming, for example, I've debated him on endogenous retroviruses. I've had many impromptu debates with him. He fails to address key points in every single debate. But then when he goes on to a channel like Answers in Atheism, he postures, but he doesn't actually address anything. And that includes the function. Endogenous retroviruses have turned out to represent important components of the antiviral immune response. Everything's important about the ERV. Your full ERVs, the LTRs are important. They're functional. Your ENV, your GAG, your POL, everything about the ERV is functional. And then your solo LTRs that I'm here to tell you are none. I'm working on a model with this. They represent functional stretches of DNA. They are not degenerated endogenous retroviruses as evidenced by their function. Regulate cell immune activation, may even directly target invading viral pathogens. You're about to see in a couple excerpts that I have that proteins that are encoded by the endogenous retrovirus, these retroviral-like sequences, can actually heighten or amplify immune responses through the binding of very specific receptors. I mean, what's going on the molecular level is incredible. The DNA is a 4-dimensional code with chock full of meta information, multiple overlapping codes. It's nested. We can read books, but we can't read books both forwards and backwards, and every other DNA letter, every other chemical DNA letter, another message. 3 messages in one, embedded into one DNA sequence, basically. And so, and these evolutionists believe that this all came about for no reason at all, by chance mostly. These very sophisticated networks, these elegant systems, all evidence of just amazing design. And the evolutionists dodge, duck, dip, dive, and dodge all over the place. The 5 Ds of dodgeball. They've owned it, I think. Notice this. One focus will be on recent advances in the role of notice recent advances. This is 2021. And these evolutionists still try and say, well, the functions being discovered, it's becoming more and more narrow. No, actually, it's broadening. It's becoming more and more wide. While many of them are usually dormant, they can be reactivated by several stimuli, including viral infections. That's what I've been saying forever, guys. We have a lot of, especially non-coding areas of the genome, like your pseudogenes, but also your ERVs, a lot of these retrotransposons. They're there, they're silent, they're turned off, they're dormant until they need to do their job. Just like we have specific ERV sequences that in development, there's these certain developmental windows where they're active and they're basically protecting the growing baby from being harmed through viruses. And once these specific developments are, they're safeguarding the baby, the growing baby. And once these developmental windows are over, then these ERVs are basically done their job, but they can also be repurposed and called upon later through other stimuli or stressors, such as viral infections. An infectious virus integrates itself, enters the host, and then these dormant retroviral-like sequences or units, they are awakened basically, and they can do their job. And so it makes sense to disregard a lot of the DNA, including your retro elements, as being non-functional junk, when you would need almost unlimited testing and sensitivity to conclude whether any of these sequences are non-functional in light of what we know is really going on in terms of adaptation and stressors and external stimuli, but also in development, these various stages of development and everything that goes into the building essentially of that fully formed baby. There's a lot that goes on. It's amazing. ERVs have become integral parts of immune defense mechanisms and help to fight off invading viral pathogens. Notice to the evolutionists, they're so biased, they're so unobjective, they don't want to question the paradigm. It's a great time to be a biblical creationist. They ask, well, why do organisms share endogenous retroviral-like sequences? I say like sequences because, again, the majority of ERVs, we have to understand, they represent solo LTRs or functional stretches of DNA. And then we show them, well, the reason why is because they function in what? Firstly, they fight off viral invaders or pathogens. They function in determining cell types, gene regulation, gene expression, embryological development, placental development. You know, I go on, and it's never good enough. Oh, well, those have been co-opted. Thank you for the story. Cool story, bro, but I want science. I'm sick of imagination. Evolutionists, they hope, dream, and imagine that whales and strawberries are related, but they don't have the evidence to back up their story or support their story, unfortunately. Notice this, receptor interference. So ERV proteins, they actually interfere with incoming viral particles. We wouldn't want to live without viruses in the world for the ecosystem in general, but we also wouldn't want to live without viruses in our body. We wouldn't want to live without viral-like DNA or units in our body. There's a lot going on. It's a very complicated, it's a very elegant and sophisticated system. And there are many interactions with DNA units like ERVs and genes and protein coding genes and non-coding genes. And the evolutionists simply can't explain it. And they also, because of their basic assumptions, evolutionary based assumptions, they can't make the kind of predictions that design advocates or creationists can. So notice this, apart from rather broad and unspecific immune modulatory activities, endogenous retroviral ENV proteins. So your ENVs are your envelope proteins, okay? And they actually function in blocking harmful viruses from entering. And how do they do this? They do this by binding to very specific receptors that infectious viruses might actually use to enter the cells of hosts. And these ENV proteins, they actually have the potential. They function to enter viruses, which as a result interferes with the viral life cycle. This is amazing stuff, guys. And this actually, it inactivates viruses before they infect new host cells. These are superheroes within the genomes of living organisms. And are we really surprised that we have antiviral protectors in humans, antiviral protectors in chimpanzees, antiviral protectors in gorillas, antiviral protectors in all forms of life? This should not be surprising. And because we share much in terms of overall phenotype or anatomy, morphology, and physiology with all forms of biology, then of course we are going to share these DNA units as well. This should not be a surprise. And the evolutionists really are grasping at straws. They really are. This mechanism involves direct binding. So notice what I just explained. Involves direct binding of cell-associated or cell-free endogenous retroviral ENV proteins to cellular entry receptors. As a result, these receptors are blocked and cannot mediate attachment of exogenous viruses using the same entry pathway. Endogenous ENVs offer a large repertoire for broadly active antiviral protectors. Everything about the ERV is functional, and everything about the ERV is necessary to carry out their roles in the first place. And so thank God, God designed them this way, not to mention exogenous retroviruses, harmful viruses coming from their hosts, coming from retrotransposons. Why would we be shocked that they have this sequence similarity then? Okay, I mean, the evolutionists, like I always say, the so-called best lines of evidence for evolution are actually some of the best lines of evidence for creation. Early reports of receptor interference date back to the 1960s, when several groups demonstrated that ENV proteins of avian leukosis viruses induce resistance to exogenous retroviruses. And this all goes back because I've seen these evolutionists try and scoff. Oh, well, God created humankind and Adam and Eve, original created kinds, in a perfect world. It was very good. So obviously there was no harmful viruses. So why did God create antiviral protectors when the harmful viruses didn't exist? I mean, way to set up life for failure. But it's called, this comes down to theology anyways, but it's called foreknowledge. The fact that God is infinite. He's outside of time, matter, and space. He's also through it. And so, of course, he designs living organisms with ways to adapt after the fall, with ways to adapt to new environments, ways to adapt to stressors or to combat stressors. So the evolutionists have bad theology and they have bad science, unfortunately. But we understand that a lot of their other functions is generating variation, generating diversity, acting again in the embryo, in gene expression, gene regulation, okay? And so many of these created units of DNA function that resemble viral-like DNA, a lot of their jobs could have been repurposed. Or they could have had multifunctionality built in, where they had primary roles, but then after the fall, that secondary role or function could have been manifested, okay? Besides ENV, endogenous retroviral. So now here's the gag proteins. Here's the remember, LTRs, gag, pol, ENV. Everything about the ERV is important. Endogenous retroviral gag proteins can also restrict retroviral replication. So notice how every component or element of the ERV is functioning in their antiviral roles. This includes the gag protein of HERV-K, which co-assembles with HIV-1 gag, thereby reducing the production of fully infectious HIV-1 particles. And so, again, these functional roles are exactly what a creationist would expect. They explain why we share these with other creatures. And the evolutionists are simply not up to date on the data, because I've heard many evolutionists say, well, it's just an aspect of the ERV that's functional, but it's not the ERV itself, the unit that's functional. And I'm showing you, no, the ENV, necessary. Your gag proteins, necessary. Your LTRs, necessary and functional. Everything. But that's like saying, well, me as a human being, you know, I have arms that are functional, or a liver and a heart and lungs that are functional, but, you know, the unit itself's not. No, the unit that I am, a human being, which comprises many different parts and components, is functional. And with the ERV unit, the components of it are functional. The unit is functional. That's why a lot of these evolutionists, like Dr. Stefan Frello, who I debated years ago, you know, he likes to comment in these Facebook groups. And he commented on my last ERV video. And I said, I'll debate you anytime on endogenous retroviruses with half my brain tied behind my back. And he was silent, because he knows he's out of date on this topic. Thus, the ability of endogenous viral gag proteins to interact, notice they're interacting with their exogenous counterparts. And so the way that they're designed, the way that they're built, the way that they're set up is necessary for these interactions, these sophisticated interactions, may confer blocks at several steps of the retroviral replication cycle. Now, a lot of your evolutionists, you'll notice Dan Stern Cardinale, he just said, well, a lot of your ERVs or ERV-like sequences, they're not transcribed. Right? DNA transcribed into RNA, translated into protein, but a lot of the genome functions as non-coding DNA and function in regulation, where we have these RNA genes, all different types of them, that are performing many functional roles. And so, as he put it earlier, some of them aren't transcribed. And this has been brought up to me in debates in the past too. But notice this. All of the antiviral mechanisms described above depend on the transcription and or translation of endogenous retroviral sequences. So we've got transcribed endogenous retroviral sequences. They're talking about translation of endogenous retroviral sequences, and these make up a lot of the functional roles. But then you got, you know, certain evolutionists, militant evolutionists say, well, what about the ones that aren't transcribed? But notice this, nevertheless, transcriptionally silent ERVs can also, notice the word also, provide a selection advantage to the host. For example, the presence of repetitive ERV elements may promote recombination processes that increase the number and or allelic variety of host genes. So what this sounds like to me is your transcriptionally silent ERVs. They have a significant benefit to the host where they can promote recombination to increase variety in host genes and make for a greater potential to fight off viral invaders and pathogens. We have functions for everything, including functions for ERVs or ERV-like retroviral-like sequences that the evolutionists say are evidence of junk because they lack certain components like GAG, POL, ENV, and they only represent solo LTRs. When these solo LTRs serve as binding sites for initiating or enhancing gene expression, transcriptionally silent ERVs also provide an advantage as they can promote recombination. And so recombination produces more variety of host genes. We have more of these ERV elements now that can actually fight off viral invaders. It's almost like adding and training more soldiers in an army, which makes that army even stronger and more equipped to take on and battle invaders. That's what this sounds like to me, and I think it's fascinating. I think we got to do more study of it. Notice this, increasing knowledge. Our knowledge about endogenous retroviral elements involved in cell immune responses is constantly increasing. How many times do we as creationists predict? Well, we predict that in the future, we're going to find more and more functions. And the evolutionist scoffs. Oh, that's an argument from silence. Oh, that's not, you're not going to be able to demonstrate that. That's far-fetched. It's like, no, these evolutionists have not read these technical papers in their own secular literature that constantly tell us that they're just in the beginning stages of understanding these functional roles. Notice, constantly increasing. Guys, more and more and more. Take us to 2030. We're going to have even more evidence for DNA function. Take us 20 years ago. We didn't have this kind of data. This is mind-boggling. Recent key findings include the discovery of retroviral ENVs restricting retroviral infection in primates. Okay, so I want to point out, guys, DNA sequences or units that evolutionists call ERVs, or the ancient remnants of viral infections. Remember the greater question that I always ask: are these really the ancient remnants of past viral infections, or are they created units of DNA function? But the evidence suggests that no, they're not the ancient vestiges of viral invasions. The evidence suggests that they were front-loaded into the genomes of living organisms as designed units of DNA that operate in the immune system. And the way they're designed is necessary for their operational roles. And then we also have solo LTRs, which represent the vast, vast majority of what evolutionists call ERVs. And yet those have their own sets of function. Do you see how this all fits together like a glove? No, it's not a weakness. It is not a limit to our model. Now, the fact that they resemble viral genetic material, as I've said many times, is because these similarities are necessary for their functional roles in mimicking and interacting with what? With real retroviral DNA, which exists. And invading retroviruses, they want to harm. They're not here to do anything that's good or beneficial. And so thank God we were front-loaded or designed with many classes of functional DNA elements that are involved in protecting us from these invaders. And remember, these roles are amplified or heightened during immune responses. And so they may be silent for the most part. They may be lying dormant until, guess what? A host, a biological organism, is invaded externally from harmful viruses. And now, thank God, we have these antiviral protectors. We have these superheroes in the genome that can be called upon or essentially manifested to protect us from those viruses. And even your transcriptionally silent ERVs can apparently, according to this paper, promote recombination, which increases variety and increases the number of antiviral protectors in the genome to fight off the invaders. It's a great time. It is a great time to be a creationist. And we're going to wrap it up with an example of why it's a great time to be a biblical creationist, because even your most militant of critics, like Dr. Dan Stern Cardinale here, who lives and breathes fighting young earth creation, he doesn't have good responses. He has no response for how these functions can come about. Yeah, they can say co-option all day. They can reiterate their story over and over again, but that doesn't make it science. These guys are bloated with evolutionary philosophy. It's evolutionary philosophy for breakfast, for lunch, for dinner, for dessert, and as a bedtime snack. The evolutionary philosophy is leaking out of their ears and nose. They've been preaching evolutionary philosophy for so long that they can't even see that they're preaching this evolutionary philosophy. So I am going to reshare, but I just want to make sure that the audio is shared. Okay, here we go, guys.
The specific question, have we directly observed an ERV that integrates and then acquires a beneficial function in the host? The answer is no. All right, we get in chapter 2, we get all this stuff about like junk DNA, ERV function, design, diversity. None of that stuff matters. Doesn't matter if they're functional. Doesn't matter if they're, designed in place. Whether they're functional or not, just 100% doesn't matter. It's all irrelevant. Okay, then we get to chapter 4. Now, most of chapter 4 is also irrelevant. It's super repetitive. So there's a lot of like, it's not junk DNA, it's actually functional. Like, I don't care, because the function thing just doesn't matter. ERVs are functional, therefore they were put there. But that doesn't matter. Doesn't matter if they're functional. Doesn't matter if they're designed. None of that stuff matters. And I want to be 100% crystal clear here. It doesn't matter if ERVs were designed. It doesn't matter if ERVs are functional. None of that stuff matters. Have we directly observed an ERV that integrates and then acquires a beneficial function in the host? The answer is no. Have we directly observed an ERV that integrates and then acquires a beneficial function in the host? The answer is no.
The answer is no.
That's all! He's down! And it is all over!
Okay, guys, I think that's the perfect place to end it. Even the loudest of evolutionists that a lot of your critics will go to for answers, those militant evolutionists themselves, they lack responses. And even they are guilty of evolutionary philosophy and just a lot of storytelling. So guys, share this around. Check the description box. I have done a ton of work on endogenous retroviruses. I am continue working on this topic. And so stay tuned for more. I am going to be doing a series very shortly. I'm currently working on it, debunking Stated Clearly. And so Stated Clearly has at least 3 videos that I know of endogenous retroviruses. And so it's going to be a comprehensive series. I am going to go through it sentence by sentence, and I am going to address what the video says. I am going to address any of all arguments that the videos advance. And so I'm looking forward to that, guys. Again, check the description box for debates I've done on ERVs. You can find my endogenous retrovirus handbook, and you can also find previous lectures and presentations. So with that, Standing for Truth is out. God bless all.